Hello, everybody. And this is Benny Yang. I'm the CSO of Biocytogen. It's really my pleasure to join BioKorea and give this presentation. And in the next 20 minutes, I will introduce to you the RMAS platform, a very cool technology to discover fully human antibody drugs. And I will use two examples to show you how we can use this platform to drive antibody research. First, a little bit background about Biocytogen. Biocytogen was founded back in 2008 in Massachusetts, United States. As of today, we have more than 1,600 employees over the world. And actually, 50% of our employees are for our research and development. At Biocytogen, we produce a large number of humanized mouse models for preclinical research, including efficacy study and the toxicity study. We also build our own pipeline, which has the first-in-class and best-in-class potential. And we also collaborate with many other biopharmaceutical companies and biotechnology companies to explore novel drugs. Today, our headquarters is in Beijing. We have facilities in Boston, in Europe, and also in Jiangsu, which is close to Shanghai. First, I will introduce a little bit about the fully human antibody. So this chart shows a summary we did a couple of years ago. So we found there's a very strong trend of using fully human or humanized antibody for clinical research. And for fully human antibodies, there's basically the two different fashions. The first one is in vivo approach using the transgenic mice. The other one is using the in vitro visual display. But when we choose antibody, we really think about two things. The first thing is the diversity of the antibody pool, whether this antibody have a larger diversity of epitope and a sequence, and we also we consider about what's the quality of these antibodies, whether they are friendly for the CMC downstream process development, and whether they can express at a very high level, and whether they are stable and they can be purified easily. For the in vivo approach, in the past, the field had developed several novel mouse models. This including the first generation transgenic mice, which has been used by BMS and Amgen. And later, Regeneron and other companies generated the second generation antibody transgenic mice. For example, the VelociMune from Regeneron, which they replaced only the variable region but retained the constant region for better B-cell development and the proper antibody response. In the last several years, Biocytogen developed new mouse models called RMAP. This mouse is a second generation antibody humanized mouse models. So they also had to replace the variable region but retain the constant region. But it's different from others. This is the only available mouse model have a complete variable region of the human kappa chain locus and the heavy chain locus. How is this achieved? So we know it's very difficult to replace the variable region of the antibody locus because it's a very large locus. For the heavy chain, it's 2.6 megabase. So in the past, people have to do step-by-step replacement of small gene fragment. But at Biocytogen, we're using a novel technology called SuperC, which allows us to replace the megabase scale DNA fragment in one step. So here, we replace the heavy chain locus from the very upstream of the V genes to the very downstream of the J gene. So we replaced every single gene of the mouse variable region of the heavy chain with the human counterpart. So this is achieved in one step. And we did the same thing for the kappa chain locus, and then we cross these mice together. And we generated these new mouse models called RMAP, humanized mouse models. So I would say by design, this is the mouse will generate identical antibody repertoire as what you were observed in human. So it should be has a high quality. So we have a complete thorough analysis of the characterization of these mouse models. If you are interested in, you can contact us. So besides that, we also consider how to generate high quality binders. So what we are talking about is about 1,400 genes. That's extracellular genes which has a druggable potential. So we pick about 150 genes from this list, and then we compare the mouse and the human gene sequence, and we make this chart about their homology between mouse and human. And we found many of these targets are highly conserved between mouse and human. So this is further illustrated here using CD73 as an example. So this protein is a very interesting target, but it has a 90% homology between mouse and human. So we colored all the identical residue with green color. You can see all the surface areas are almost colored with green. Only a few residue are not identical between mouse and human. So if we immunize a mouse with human CD73, it's very hard to generate diverse binders because the immune tolerance exists in here. So what we can do is actually we come up with a strategy is to knock out that particular gene on the randomized background, and indeed we found in terms of the antibody titer and also the antibody binder, the number are much increased. So we use the beacon for routine antibody screening. So without the knockout, using wild type randomized mice, so we usually for target A, we can only generate 100 binder. But if we knock out that particular target on randomized background, so this time we can generate 600 binders, six-fold increase over the non-target knockout mice. So this holds true for many different targets. So on this figure, all the target have the sequence homology between all mouse and human between 53 to 94%. So that's all we come up with the randomized hits campaign. So this campaign is really is intended for generate high quality binder for over 1,000 extracellular targets. What we do here is for any gene we want to generate antibody, we first knock out that particular gene on the randomized background. Then we do the immunization. So without the knockout, you can only generate those yellow antibody that's recognize those non-conserved epitope. But if we knock out that particular gene, then we can generate not only the yellow antibody, but we can also get the red antibody that's recognize those conserved area. So we'll get more binders. And also many of these binders, since they can cross relative to mouse and human antigen, so we can, using the wild type animal for the downstream screening, using all the existing in vivo mouse models for cancer, for autoimmune disease. So this will make the research and the antibody screening or validation much straightforward. Once we have a good candidate, then we can, using the same antibody for the further preclinical study. So besides fully human antibodies, we can also generate bispecific fully human antibodies. So here I just show some examples of some typical bispecific antibodies, 1+1 format bispecific antibodies in the clinical study. So these bispecific antibodies, they can bind to two different epitopes on the same target, like as on the top left, which are the bispecific antibodies, or we call biparatopic antibody, recognize two different epitopes on HER2, or they can bind to two different tumor-associated antigen like c-MET, EGFR, or they can even recognize two different cell types, like here for T-cell engager. So one arm can bind to CD3 and the other arm can bind to a well-known tumor-associated antigen. So this kind of 1+1 format antibody, bispecific antibody are very useful. But how to generate these antibodies? So one thing we need to consider is how to make really avoid heavy chain and light chain mispairing or heavy chain mispairing. So for heavy chain mispairing, this can be easily solved using the knobbing two-hole approach. But the difficulty is to address the heavy chain light chain mispairing issue. So here we come up with this common light chain solution, and here using based on the RenMap mice, so we generate this mice called RenLite. So in this mouse, the heavy chain still have the same repertoire as the RenMap, but for the kappa chain, instead of replacing with the human counterparts, we used a fixed, rearranged human kappa chain sequence to replace the entire mouse variable regions. So in this mouse, all the antibody or all the B cell will have the same light chain sequence, but they have different heavy chain sequence, which will give them the diversity of the repertoire. So once we fix the light chain and we immunize these mice, and we can generate many binders. And here just show two different examples. For each example, we can generate more than around 1,000 antibody clones. And we analyze the affinity of this antibody clone using the SPR technology, and we find they all have reasonable or very desirable affinity against the particular targets. So most antibody have the affinity around the nanomole range, and some of them even have higher affinity. So this is really ideal for the antibody drug development. So here I will show you two examples to how we can use these RenLite mice to generate novel drugs. The first example is bispecific antibody against targeting intratumor Treg cells. So Treg cells express many markers, but these markers are also shared by activated T cells. So how to target these cells specifically? So we think we can pick two different markers highly expressed by Treg cells, and we can combine them into one molecule using the novel two-hole approach. So this molecule will target these intratumor Treg cells better than the activated T cells. So we built this molecule YH006, and here just show the purity and the stability of this molecule. So just after one single step of purification, so we're using SEC, we can see a homogeneous fraction. And then we treated this molecule at a 40-degree high temperature for different days. And you can see either by SEC or by HIC for hydrophobicity, or using the ICIF to show the charge heterogeneity. This molecule is very stable at the storage condition. And further, we showed in vivo this molecule 1+1 format bispecific antibody is more potent than the parental monoclonal antibody. So this is the MC30A syngeneic tumor mouse models. So we using humanized mice expressing both target A and target B, and we implant tumor, and then we give it 3 injections, and we showed that bispecific molecules can completely inhibit tumor growth, and which has much better efficacy than the parental monoclonal bivalent antibodies. So the other example I want to share is about how we can use this technology to build better ADCs and antibody drug conjugates. ADC is becoming a hot area in the last several years, and more ADC drugs have been approved by the regulatory agency. And ADC not only targets the blood malignancy, but they can also be effective in treating the solid tumors. And many ADCs using the MMAE payload, but also different payload have been shown success in the clinical trials. But the one problem of ADC is that you have many tumor-associated antigens are also expressed by normal tissues. So this will cause the off-tumor toxicities, which prevent the people using the higher dose for treatment. So the question we want to address is how to improve the tumor cell selectivities. We think a bispecific solution is also works here. So we think many tumor cells express a combination of tumor-associated antigens, and these antigens usually express at a low level or not expressed very well in the normal tissues. So instead of using monoclonal antibody, if we design a bispecific antibody, recognize 2 different tumor-associated antigens, and this molecule will bind to the tumor cell much better than the healthy normal cells. And if we link the small molecule drug on these antibodies. So this ADC will target more efficiently to tumor cells. But besides that, we think also when we target two different receptors, this usually causes the clustering of the receptor on the cell surface, and which will drive the internalization of the whole molecule into the intracellular compartment, and then later get degraded in the later endosomes. And this is really ideal for release the payload into the tumor cells. So this example I want to show is called YH012. This is a bispecific ADCs targeting HER2 and TROP2. In this molecule, we still using the MMAE as a payload because this payload has been shown success in 5 different drugs being approved. And here we not only we studied HER2 TROP2 ADCs, we're also using the parental HER2 antibody or TROP2 antibody as a control, and we're also including the HER2 monoclonal antibody or TROP2 monoclonal antibody, monovalent antibody as a control for comparison. So we think this bispecific ADCs will target to the tumor cells because the tumor cell will express both HER2 and TROP2 at high level, but the normal cell will express very little. So this slide shows the stability of YH02 either by SEC or by HIC or by ICIEF. So we store the molecule at 40 degrees for 7 days, or we treated this molecule by repeated freeze thaw, or we treat it with a low pH. We show this molecule is very pure, and also it's very stable. And we show the bilateral transfection into the CHO cells. This molecule can be expressed at a high level, over 0.4 milligram per ml. And when we conjugate with MMAE, we check the profile using the HIC. We see this is very typical to the conventional monoclonal antibody. So means the MA conjugation works very well. So we chose for in vitro study, we chose the cancer cell line called NCI-N87, which express both HER2 and TROP2, shown by RNA-seq analysis. And here we did real-time endocytosis analysis, and we showed that YH012 is endocytosis much efficiently than all the other controls, including the parental TROP2 HER2 antibody, or monovalent antibody, or TROP2 benchmark, or HER2 benchmark. This data is also illustrated in the right panel using a different way to express. For in vivo study, we're using the NCI-H9775 lung cancer cell line, and this cell line expresses HER2 and TROP2. And we implant this cell line into the immunodeficient mice, BNDG immunodeficient mice, and we give these mice 2 treatments. And we show the YH012 MMAE completely suppress the tumor growth. And so the suppression is much better than the parental monoclonal antibody, monovalent antibody, or the benchmark for HER2 and benchmark for TROP2. So here I think because of the time limit, I just want to give a quick summary. So I have shown we can using the Zenmab or Zenlide mice and to generate the high quality antibody. If we knock out that particular target first, and we'll get even more binders and which have more diverse epitopes, and we can assemble this a bispecific antibody using the sequence from 2 different targets. So the assembly is easy because the light chain has the same sequence. So we only need to using the knobbing tool approach to solve the heavy chain pairing issue. Because this molecule is very stable and it's pure, we can further conjugate this bispecific antibody with a linker and payload, and we can using this bispecific ADC for in vivo and in vitro studies. Also for the mice campaigns, so this is a campaign to generate the screening the antibody drugs for over 1,000 druggable targets, extracellular targets. So we launched this campaign 2 years ago, and you can check the updates on our website here, the www.zenmab.com. So many of these many of these targets, we already have the generated start immunization, and we even have the antibody hits. And if you are interested in, and I think this will be a good start for your research. And for the business, I think we have been collaborating with many different pharmaceutical companies, and I think they either can license our technology or they co-develop the assets with us. I think for the win-win purpose. I think the last thing, I want to thank everybody for your attention. And if you are interested in and want to have more information, you can check our website or contact our BD and just show us email, and we'll get you in touch. Thanks very much for attention.